behavioral mechanism.
Sorry, go ahead.
How neurobehavioral mechanisms of social prediction and attunement comes about from different perspectives. We're really happy to have a discussion today. We'll take on some questions after every talk. Feel free to raise your hand and we will try to see it and get your question. With Simon's talk, she can answer questions after David's talk at the end of the session. So I'll start right by because I'm the first speaker here. So let me share my screen. Do you see my first slide? Okay, good. So let's go right to business and look at this. We see two kids that they seem to have a common goal, and that is to synchronize. And look at where their gaze is directed at. They're looking at each other's gaze. They're looking at their partner's body and they adjust their own. I can tell you that these two are siblings. They know each other very well. And when they get in the zone, there they are. This is almost rewarding on its own. It's really fun to look at, and it also seems to be nice for them as well. Now you can clearly see that these two are social animals that know how to synchronize with one another. And I say this with pride because these are my children. But I think really that looking into the neural and behavioral mechanisms of social synchrony, of what it takes to do this, what they just did, can be really informative to study how we bond and how the brain is using the information around it to maximize metabolism, basically. And when I say metabolism, I mean that in social animals, the evolutionary plan really does not only rely on the physiology of the individual, but it also takes under consideration the presence of other conspecifics, of other individuals from the same species. If we think about this as a definition for a social species being dependent on another conspecific for metabolism or physiological regulation, we see that all birds and all mammals are social or dependent, at least at some point of their life, because newborns cannot regulate their own physiology. And they completely rely on a dedicated caregiver to regulate for them many aspects of their ongoing physiological processes. And this is a term I call allostasis. And this includes their energy expenditure, their immune function, the temperature, the autonomic nervous system, and basically a lot of features of their physiology. Now, in order for this to go well, the infant must be equipped with a system that can sense fluctuation in their physiology or in their allostatic demands. And this system, which we call interoceptive perception, needs to be connected to a motor system that signals out these fluctuations to the environment. And so that someone else can come and take care of these fluctuations for them. Now, we can think about how to map these social signals of regulation using this two-dimensional space consisting of valence and arousal. This is the affective circumplex that was presented by James Russell a few years ago. And we can use this model of affect to try to systematically model social signaling of regulation that infants sense. And the way we do it is that think about the horizontal axis, the valence axis, as moving leftward on this axis can be a negative allostasis deviation, and moving rightward can be regaining allostasis. The arousal axis, the vertical axis, can be thought of as the amplitude of deviation, such that negative allostasis deviation can have a mild amplitude, and it can have a very dramatic amplitude. And the same is true also for the right side of the space. Now we can model here, we can model, wait, I don't see, okay. We can model how infants signal out the regulation needs throughout interaction or while they interact with other people. And we can also model how the parent is responding. So we can do it simultaneously for two people and more at the same time. Now here, this is real data. I'm going to press play, so we're going to have time here too. So this is real data of a mother and an infant interacting while we code their the regulation cues that they spontaneously communicate during the interaction. You can see that both are happy on the right side, and here the infant becomes dysregulated. They start to cry. Mommy is not synchronizing with him. She stays on the positive side of the space and tries to bring him back to the positive. There she succeeds. Now he's going to calm down. See, he's going down on arousal, still staying positive. We can do this for every kind of interaction and get a lot of data. We can ask, for example, questions about infant temperament and parental care, and ask which infants spend more time on this side of the space compared to this side of the space. We can ask about the neural, the physiological features or components that are associated with different strategies of parental care and with different regulation patterns in infants. And we can also ask questions about development. So what is the most optimal developmental pattern for infants with different outcomes? Parents have to be very attentive to their infant cues. Otherwise, infants would be dysregulated. They cannot survive unless someone is super attentive to their cues. And think about it, it is very hard to regulate physiology in another person. When we are in pain, our brain gets an internal signal about it. When we are hungry, when our glucose levels go down, we get a signal about it. Our brain knows it. But when we need to regulate someone else's glucose levels, we need to be super attentive to very subtle cues that this human is sending us in order to regulate it in the temporal resolution that we need, that we want. So what we test in this experiment that I will show you is the neural mechanism in parents that helps them detect these subtle regulation cues. I hope that you will have the sound now. I'm not sure. Do you hear the sound? This is Shira. Shira is playing in her house, and you can see here the coding for with our system that she was given. Now, when Shira is engaged and playing, you will see a positive deviation, and when Shira is frustrated, you will see a negative deviation. Frustrated. Now you see that these cues are very, very subtle. But to Shira's mother, who is right now lying in the scanner, these very subtle cues are actually very informative. Shira's mother is relying on these cues to get information about Shira's well-being moment to moment. And so this is our experimental setting. We have a mom lying here in the scanner. And in this experiment, we use a special scanner that can combine fMRI and PET during the same scan while the mother is watching a film of her infant. And what we tested is whether we quantified the amount of regulatory signals that each baby communicates during a spontaneous 2-minute video. And we then wanted to see whether the regulatory cues that the infant sent can elicit a dopaminergic response in mothers and where. And what we found is that when mothers watched videos with a lot of regulation signals here on the x-axis, she secretes more dopamine that was measured here using PET in the nucleus accumbens. You can see it here. So the nucleus accumbens, the PET secretion in the nucleus accumbens was increased with the amount of deviations that the infant signaled during the video. Now, this region is not working alone. The dopamine secretion in mothers is associated with the recruitment of a whole network, which we actually know from research in human and non-human animals, that this circuit that includes the amygdala, the medial prefrontal cortex, and of course the nucleus accumbens as well, and also the hypothalamus that secretes oxytocin to regulate the dopamine secretion that we just discussed. In this circuit that is very consistently involved in maternal care across species, but it's also not specific, of course, to maternal care or even to social behavior. So this is a circuit that supports any kind of motivated behavior, but it's very, very persistently associated and demonstrated mechanistically in social behaviors across species. The more oxytocin and dopamine are secreted within the circuit, the more maternal behaviors the mother would perform with her pups. And in humans, we also associated this circuit, the connectivity within the circuit, the dopamine secretion within the circuit, to how synchronous the mother is with her infant. So to how attuned the mother is to the infant cues and how well she can adjust her own behavior. So if we have this neural circuit that is very persistently associated with social behavior, does that mean that as a social animal, infants need to be equipped with this circuitry at birth in order to survive as a social animal? And when I ask this question sometimes at class, though, I mostly get the answer yes, but actually the answer is no. When we look at newborns' brains, we see that the circuitry that we know is associated with social behaviors in adulthood is not traceable, at least not in FRI, in human newborns. We do see intact in newborn's brain is sensory and motor control networks, but they are separate. You can see that each sensory modality is actually separated from the other one. And what we do not see in newborn's brain are two super important features. The first is long-range connections that connects distal parts of the brain. And we also do not see multimodal integration. And multimodal integration is super important to the point I want to make about the brain as a prophet. You see that multisensory convergence, the ability to experience the world as convergent and meaningful, only develops after a few weeks, and it is not genetically predetermined. So it emerges, depends on experience. When infants experience multimodal information together, they start to associate it with time at approximately a few weeks of age. And here, this is a model that can potentially explain how multimodal associations are generated. So in this diagram, each ring represents a different type of cortex, going from very laminar cortex to less laminar cortex in the inner circle. So different type of cortex have different levels of lamina, of lamination. And we see that the primary sensory cortices, here the V1, the auditory one, the somatosensory cortex one, they are all located at the most laminated kind of cortex. And at first, perception is being perceived in a unimodal way. Then information is projected to association cortices, but at first the association cortex is still unimodal. So you see here visual association cortex that integrates information from different visual inputs. But that information is being projected to a higher level in the hierarchy to multimodal association areas. Now, these multimodal associations are very useful for two reasons. First, they enable to perceive the world as convergent, but it is also useful because it helping the brain to predict. Because if the brain is receiving input from a visual source here, they already have some sort of knowledge about what's going to happen in this channel and in this channel. So multimodal association is the first mechanism that enables the brain to predict at first across modalities. But when sensory information is associated, it can be internally represented. And if we go back to the terminology of psychology, we can call it a concept. So when multimodal information is associated in association cortices and internally represented, we can call it a concept. And concepts are very useful for prediction. Let's think about an example. So all four-legged animals that bark are dogs. It is a very variable population. They can be very big, very small. They can be blonde, black, white, but still pretty easily we can categorize them into the same category and represent them in a much more abstract way. So with much less information as a dog concept. Now, the dog concept is a super useful predictive probabilistic model because I already learned, I know about dogs. I've learned about dogs, and I know that if I see a dog, I know how it would sound. I know how it would behave. I even have predictions about myself. If I like dogs, I know that I would want to approach and touch them and pet them. If I dislike dogs, I know that I would have a stress response. So this concept is helping my brain to predict both in time and in space. And the brain knows how to organize very rich populations of sensory information into very useful constructs that are abstract, that contain a lot less information, but are very useful in prediction. Now let's go back to this little one and think about this little infant as a model-free and a concept-free creature, just because she didn't still have the experience, the sensory experience to form any valid predictions about or any concepts about the world. And within a few weeks, this creature is interacting with some social experts in their environment, and they become experts. They know how to categorize, they know how to conceptualize, and they can use this concept to interact and to regulate themselves. And how might this happen, this developmental process? And I want to suggest that the infant metabolic demands are a crucial feature to determine this development of learning to form concepts and use them to predict. And the reason for that is that the survival dependency, when an infant completely depends on their caregiver in order to survive, this produces a situation in which the infant is repeatedly exposed to social information, mommy, which is by definition always coupled with the ultimate reward, which is the regulation of allostasis. Every time the infant is experiencing any deviation from allostatic balance, in cases where the parenting is beneficial, the parent would address this and regulate the infant's allostasis. Now, this can cause three really important things. The first thing is that social information becomes more and more salient and more rewarding as days go by, and the infant bond with the caregiver. The second thing that happens is that infant learns, like any other social animal, that the most efficient or maybe the only way to survive, to regulate my own allostasis, is through social interactions. And the third thing that happened is that infants become more and more motivated to learn social concepts because they're the most rewarding concepts that exist for them at first. Now, for example, a social concept can be the mommy concept. It can similarly be the daddy or any other caregiver. At first, the infant doesn't have any concept, so they're born, they have no idea, not about mommy and not about even how to associate different sensory inputs. But after repeated care, the infants learn to associate the mommy's auditory input with the mommy's visual input, with the mommy's tactile input, and so on. This information is gathered in association cortices, but it is also associated with a different source of sensory input, which is the interoceptive source. And every time the exteroceptive source about mommy exists, also there are interoceptive consequences to that. So the mommy concept really associates exteroceptive information about social features to interoceptive information about my own well-being. And if all goes well, or infants that are being raised with a caring caregiver, will very quickly form a concept by which every time I hear mommy's voice, I already have predictions about my upcoming changes in my glucose levels and can start to secrete insulin, for example, and so on. So think about it. A newborn by the age of 2 or 3 days already have dozens of trials linking social information to the ultimate reward of regulation. And this is a very efficient learning regimen. So just like the dog concept, more abstract concepts can be learned through the same mechanism because, for example, emotion concept, every time an infant feels an array of feelings within their body and in the context of the environment, the parent can regulate this, can organize these sensory inputs and connect it to a word, fear, happiness, sadness. And this is how infants learn to label an array of sensory inputs with more abstract a word, really, which is completely meaningless until it is associated in a significant way to the way I'm feeling, to my allostasis, and then it becomes imbued with value. And of course, this has also very useful predictive values because once I can organize my feelings into a concept, I know what's going to happen. I know what I prefer to do. I know what's the environment is going to be like, and so on. And if we look for the last few seconds at the brain again, we inspect the neural hubs that are considered to be the most central social regions in the brain. In this example, it's the anterior insula. If we look at the anatomy of this region, which is really considered a hub for social processing and for affective processing in the brain, we see that this region is connected to every kind of primary cortex in the brain. So maybe what this region is doing to be a social region is really just multimodal association between different modalities. Here we see the ectoreceptive modalities, but this region is also connected to the posterior insula, which is the primary interoceptive cortex. And really, I hypothesize, or I want to propose that maybe this region is social simply because it's organizing, it's performing multimodal association from interoceptive and exteroceptive inputs. And it's not only the insula, we see that social and emotional processing in the brain, generally, if we want to make a general rule, they persist of regions that regulate allostasis in the body through the HPA axis and through the autonomic nervous system. They are connected to sensory motor regions that, of course, sense the information and organizes action. And these are all associated in association cortices in different kinds of the brain. And we propose that this might be associated and connected through experience or very much affected by experience as infants are constantly cured by. And with this, I would end. I would thank my lab and you for your attention. Thank you.
We have time for just a few questions. So if anyone wants to ask something, now's the time.
If I can.
Sure.
First of all, thank you. Fascinating. So according to the theory you have proposed, if I understand correctly, we expect to see developments in the multimodal cortex like very early, like after a couple of even days, if I understand. So I wonder if there's any evidence for that.
I think in human infants, it would be really hard to measure, but theoretically, I think, yes, you're right. We should be seeing change after a few hours. I mean, even before birth, we start to get multimodal integration in utero with specific modalities. But I think what's changing after birth is the regimen that associates the social information with the interoceptive information. So in utero, the interoceptive regulation is ongoing. You don't become hungry. And if you're in pain, you're not regulated in a way that is consistently associated with the caregiver. So what changes after birth is the learning regimen, is the association between the social and the interoception. And I think we should be, if we would measure it in an optimal way, we should be able to see traces immediately.
Okay.
Okay. So now we're going to have, wait, let me, okay. So Simon Shammai-Tsuri is going to give us a talk about interbrain coupling underlies mutual predictions in social interactions.
So I'm going to share Simon's video, and she's here to answer questions. Oh, Simon, go ahead. Everybody sees my screen now? So
hi, everyone. Thank you for organizing this remarkable symposium. Today, I would like to share with you a few insights from our lab about interbrain coupling, basically how interbrain coupling underlies mutual prediction in social interactions. First, I would like to thank the student that participated in the experiment, my support team, and of course the funding agency that supported the experiments that, the funding of the experiment that I will present here. I will start with a very impressive video clip of herding sheep. Sheep live in herds as a form of protection. They move from one fertile grassland to another in a group in this very harmonic and synchronized manner. And you will note that it is not only fascinating to participate in activities that involve joint movement, like when we sing together or dance together, but it is also fascinating to observe synchrony. Now, remarkably, this fascinating behavior of collective action is not unique to sheep. We see schools of fish, flock of birds, and of course, in many, many human action, we see joint synchronized action that involve alignment, like when we dance together, when we play sport, when we converse, and even when we walk together along the street, we synchronize, we tend to develop this synchronized pace. And notably, it is possible to observe more higher level of synchrony in human behavior, not only alignment in movement, but also alignment in emotion. For example, when we share the sadness of a friend, when we share the joy of peers, we see this emotional contagious behavior that involve another form of alignment. In addition, we see alignment in cognition, for example, during social conformity, when we align our beliefs, our thoughts with someone else. In a recent model, we suggested that we can view perhaps synchronized movement, emotional contagion, and social conformity as interrelated process that rely on a shared neural network. So synchrony, motor synchrony, cognitive synchrony, and emotional synchrony that has seemingly different behavioral manifestation are perhaps linked to one another and are supported by same neural network involving at least 3 basic neural network: the reward system, misalignment distress system, and the observation execution or mirror neuron system. And so we view the reward system as a system that is responsible for signaling reward when we align with someone. The distress system operates when we observe a gap between ourselves and other. And the mirror neuron system, or the observation execution system involving the inferior frontal gyrus and the IPL, IFG and IPL, are responsible for mimicking an alignment with someone else. We may use the framework of the predictive coding approach to understand this behavior. So according to the predictive coding framework of alignment, the brain constantly generates prediction on alignment based on previous information about alignment. So we have those priors, and we expect all the time to be aligned with someone else. And once we detect a gap between ourselves and someone else or interaction partner, we operate the alignment system in order to achieve an alignment. When alignment is achieved, when there is no gap, there is reward signaling, and we keep our predictions. Now, this very simplistic model of alignment is probably more complex, since during real life social interaction, both interaction partners operate in the same manner and perhaps create mutual predictions. So the systems are not only active in one partner, but perhaps are coupled in their activities between partners. And the question is how to measure it in the lab and whether indeed those systems are coupled between participants. And a new approach in neuroscience, in social neuroscience, termed the two-brain approach, suggests that if until now brain was regarded as a one unit involving multiple networks, perhaps we should look at social interaction as an extended network with multiple intra-brain connections and inter-brain connections. Intra-brain connections are probably mediated by physical connection when we talk about functional network, but we also have functional network that are between brain, and these functional inter-brain connections are probably mediated by sensory input. And hyperscanning methods such as dual EEG, dual fMRIs, dual fMRI allows measuring brain activity from two or more individuals simultaneously, allowing the assessment of the bidirectional information flow between interacting individuals. And recently we used this approach to measure brain-to-brain coupling during empathic interactions. We scanned with EEG a romantic couple where one of the partners was inflicted with pain and the other was instructed to either hold the hand of the participant or just be present in the room. And we found that in this condition, compared to a control condition where there were no hand holding, we found increased brain-to-brain coupling. Each yellow line here represents a significant correlation in brain activity in the alpha mu band. And remarkably, this brain-to-brain coupling significantly predicted pain reduction in the suffering partner. So basically, we see that this approach allows characterizing those intra-brain networks, and also it predicts outcome of relationship. So in a recent study on motion synchrony, my graduate student, Yuval Martin Alper, carried out a study in which she wanted to examine interactive improvisation of three-dimensional movement synchrony and how mutual prediction may underlie this behavior. So she examined interbrain synchrony during free improvisation of three-dimensional movement. She recruited 80 participants that were randomly divided into dyads, and were, as we can see, they were scanned with fMRIs while performing a hand movement task with a high temporal resolution gaming device. You see that they are holding this joystick that allows tracking precisely the hand movement. And we use the time varying method to calculate the level of dyadic behavioral synchrony. And basically we had through simple condition, free movement condition where they just move their hand freely, and an instructed synchrony condition where they were basically instructed to move their hand together in synchrony. And this is a demonstration of their performance. So we developed a way to track their movement. We see that this is the instructed synchrony condition. They attempt to move their hands in synchrony, and we are able to calculate the level of synchrony in each millisecond. So we see in a picture of one couple and the level of synchrony throughout the task. So we were able to quantify level of correlation values based on motion velocity direction. In the x-axis, we see the representation of time. The y-axis represents the time lag between participants, where zero lag is here represented in the white line. And the red lines marks basically synchronization periods. So we see that there are minor synchronization period in the free movement condition and longer synchronization period in the instructed synchrony condition, higher level of correlation represented with the yellow. So this is basically a validation of the task. We used the FNIRS and we organized the channel placement above the inferior frontal gyrus and the dorsomedial prefrontal cortex of our participants. So we defined those regions of interest as inferior frontal gyrus and dorsomedial prefrontal cortex. And we measured coupling between those regions of interest between participants using the WTC measure, which is usually used to measure brain-to-brain coupling in FNIRS. Behavioral synchrony measurements confirm increased level of behavioral synchrony in the instructed synchrony condition compared to the free movement condition. And we also created a control condition of pseudo group, which is basically a method that in which we reshuffle all our dyads and pair participants that did not play together. And this is a method that allows ruling out basically condition that there is synchrony that is not related to real synchrony. So we see no synchrony in pseudodyads, but higher synchrony in instructed synchrony condition. And remarkably, we did not find significant synchrony in the free movement condition, suggesting that this three-dimensional task does not allow the emergence of spontaneous synchrony, at least in our measures. When we look at the brain-to-brain synchrony across all channels, when we average all channels, we see increased synchrony in the free movement and the instructed synchrony condition higher than the pseudo-synchrony. And this basically suggests that our results are indeed reflecting brain-to-brain synchrony and is not an epiphenomenon of doing the same thing at the same time. However, we do see that the instructed synchrony shows higher level of brain-to-brain synchrony as compared to free movement. And when we divided our sample, our channels into region of interest and calculated all combination of brain-to-brain coupling with the region of interest, we see three significant combination after correction for multiple comparison. So we see a higher level of synchrony in instructed synchrony condition in the inferior frontal gyrus and combination, and also right IFG and dorsomedial prefrontal cortex, and also dorsomedial combination. We also confirm this finding with the brain and behavior correlation. We see that higher brain-to-brain synchrony predicted better performance in the instructed synchrony task with the significant correlation. This is the orange line. Increased brain-to-brain coupling significantly predicts performance in the task in the IFG combination and IFG dorsomedial prefrontal combination. So we conclude that overall our task is valid since we see greater behavioral synchrony in the instructed versus free movement condition. We also observe interbrain synchrony in real as opposed to pseudo-dyads. And finally, our more selective analysis of specific channels show that we see a higher level of brain-to-brain synchrony in the right IFG, right IFG combination and right IFG dorsomedial combination. And these combinations also predict behaviors suggesting that this coupling between those systems support the ability of the participant to synchronize their actions. We further confirm the role of IFG synchrony in a more basic two-dimensional movement coordination task. This is a task of 2D movement, but each participant receives, is assigned to a colorful circle. One is blue, one is red, and using the keyboard, they move their circle in synchrony. And again, we found coupling in the IFG that developed over time in the instructed synchrony condition as compared to the control condition. Finally, in the final experiment that I will present, we thought to examine whether group membership plays a role in those mutual prediction tasks. And to do so, we recruited in-group and intergroup dyads. So basically, we compared the performance of mixed group consisting of Israeli Jews and Israeli Arab that define themselves as Palestinians, and compare the performance to in-group dyads that they were composed of Israeli or Palestinian. And the behavioral, here in the upper panel, we see that the behavioral results shows some difference in the third trial, indicating that in-group dyads perform better or improve their performance over time in the task. However, intergroup dyads do not show this performance. And we see a very similar trend and even more pronounced pattern of finding in the brain-to-brain coupling in the IFG. Again, we see increased interbrain coupling. There is a gradual increase in their coupling throughout task performance, but only in group dyad. And this improvement is not observed in intergroup dyad. And this basically suggests that group membership, when we perceive our interaction partner as dissimilar to us, we see a reduction in the ability to synchronize during the task. So if we go back to our model, we see that those models of prediction that operate in one brain during real-life social interaction, we see coupling in the activity of this system. We found a coupling in the observation execution system between participants and also in the dorsomedial prefrontal cortex of participants. Of course, it is very difficult with fMILs to track the activity of the reward system. So perhaps in a future study, we can examine it with fMRI. In future direction, we also plan to examine whether it is possible to train synchrony to overcome those barriers of group membership, and also train participants that show some difficulties in synchrony. And finally, I would like to encourage other groups and other labs to use those real-life portable systems that allows tracking behavior and interactions in more ecological manner. And I would like, again, to thank my excellent student and the funding agencies. And thank you for listening.
Well, that was a super excellent and interesting talk. Thank you, Simon. I think that Simon would take questions after the session.
I can take them now.
Oh, great. Okay.
I'm available now. Yeah.
Okay, perfect. So any questions for Simon? People are shy today. I don't know why. Okay. Okay, someone raised their hand. Wait. I don't see. Okay.
Talia has a question.
Yeah, go ahead.
Talia, you're muted.
Can I ask a question?
Yeah.
Yeah. Talia? Okay, maybe we should. Oh, okay. Ken, and now we hear you. A quick question, because we are a little late in time. Toda. Go ahead, Talia.
I have a question related to episode of the use.
Talia, we can't hear you, so maybe you
I
can.
can't really hear the question.
Yeah, so maybe we can postpone it to later if we have more time at the end, okay?
Okay, so Talia, if you have a question, just send it to us in the chat. And in the meantime, okay. In the meantime, I'm going. Wait. Do you all hear this sound?
It's an exercise in prediction. Simon, you
Yeah.
can answer whatever you
So,
want.
but Talia
Let's
is
see if you
muted, so I don't know
predicted
why
the
it's
question.
happening. Okay. I'm trying to mute her. Okay, now I've succeeded. Okay, so moving on to our third speaker with some technical issues. So Yuval Hart is going to give us a talk about early warning signals in motion inference. Go ahead, Yuval.
Hey everybody, can you see my slide? Okay, so for those who don't know me, my name is Yuval and I'm a faculty member at the psychology department at the Hebrew U. And I want to talk today about social motion and how we predict during decision making other people's actions. I'll say just a few words about my group. We're a new group at the Hebrew U. So basically, today's with the big data we have, we can see both the brain and the mind activity in super high resolution, both at the neuronal level, groups of neurons, areas in the brain, connections between networks, behavior, and computation. And sort of my hope is to try and bridge these different levels. But starting, usually people think about bottom up. We'll understand the neuron, we'll understand the group of neurons, then areas in the brain, networks, behavior, and the computation behind them. I usually like to start the other way around, so understanding the computation, behavior, and then launching into the biology of things. And the basic questions that we ask in my group are, what are the computational building blocks that actually sort of compose human thought? So if this is a Lego Einstein, I want to understand the bricks that make each and every cognitive process. And today we're going to talk about social motion. So a short quote from Einstein saying that since our inner experience consists of reproductions and combinations of sensory impressions, the concept of a soul without a body seems to me to be empty and void of meaning. And this is sort of the guideline in these kinds of work. So from our body, from our movements, can we infer what's going on inside our mind? And so today I'm going to talk about early warning signals in motion inference. And actually the real question is how do we infer the intentions of others in competition scenario? And this work has been done with Mariam Waziri Pashkam and Maha from Harvard. Okay, so prediction, hopefully by now you are convinced that prediction is important, but sort of sports brings it brings it to the extreme. Let's see if I can show you. Yeah. So here's England versus Italy, the quarterfinals of the 2012 World Cup. And
desperately trying to put off Andrea Pirlo, but he's been the best player on the pitch today. Oh my word, that's one.
So Joe Hart, the goalkeeper for England, jumps too early, does not recognize the signal, and Andrea Pirlo, a soccer genius, kicks this dardale kick and makes a goal. And
Buffon goes. Torino.
So again,
Very calmly done.
Not getting
Absolute
drive.
delight for the Atzil.
And now this is the deciding kick as a goalie, Buffon, also an Italian football player legend.
Ashley Cole has his penalty saved by Buffon. Shoots.
I'm sure the celebration.
England eliminated.
Have the
And the Azzurri, after dominating the match itself.
So Italy won. So there's if you manage to predict correctly, you're a national hero. And if you did not, then not good. I'll skip the other film of ankle breakers, but it's also fun to see where people do not manage to predict in time. Basketball players, they do not manage to predict in time when a certain player changes his direction, and then they fall. And it's part of like it's the greatest humiliation you can get in professional basketball. So the fine line between a national hero and being mocked by everybody is a prediction. So can we understand better what are the underlying mechanisms that consist this great human ability? And I want to say in general, looking at it from a computational viewpoint, it's the whole field of social cognition is super fascinating just because of that. So the computations are super hard, right? These are things that machine learning, with all its might and glory, cannot perform well. So it's super hard, and yet people are super good at it. And this means that there are some computational constraints that can allow us to infer the mechanisms, hopefully in a fine-grained way. So here's the experiment. The experiment sort of mimics this striker goalie scenario of penalty kicks. So two participants, both of them are with their finger on this magnetic. Let's see if I can. So they're sitting with this pad, and the striker needs to go either to the right or to the left to get secure in her headphones, and the goal of the goalie is to reach the same position as fast as she can. Okay, so 14 pairs, 5 to 7 sessions, 30 trials. Each session, and we have sensors on the striker. We have 7 sensors, and on the goalie we have 1 on the finger. And what we see that almost between 120 milliseconds to 150 milliseconds after the attacker, the striker, started to move, the blocker starts to move as well. And this is super fast, right? If they don't see, I'll show you in a moment, but if they don't see the attacker, but only see a dot portraying the trajectory of the finger, it takes people 300 milliseconds. So between 2 to 3 times faster when you see the entire body of the attacker. So hopefully it sort of signals that there are some kind of cues that come even before motion onset. Okay, already when the finger is static, some indication of movement is there. And you can do the same with not with real interactions, but seeing a video on a screen. So this is Mariam, and she's going to go either to the right or to the left. And we see that even with videos, compared to the dot experiment, where there's just the dot going on the same trajectory on the screen. So people react earlier, even if they don't see the head, if they don't see the torso, or just the head, if they only see the head but with sunglasses, so there's no gaze. And here it's already hard to tell. But there are many cues all around, spread all around the body, that sort of signal something is coming. So, and this is another cute experiment. You can also do the following. So you can take the video, mark the frame where motion started, and then remove all the frames before motion onset, and replace it with a still frame. So the first frame is just copied throughout until motion starts. Okay, so this is the full video. This is the cut video, and again, you see that there is a marked difference in response time, whether they see the motion before it actually started. So you see the person sort of fluctuating before they start, and the actual video. Okay, so hopefully I convinced you that there are some signals coming before the motion itself, but can we find it? Can we know what they are? So in order to do that, we thought that it could be interesting to think about this motoric decision-making, going right or left, as a bifurcation event. Bifurcation events are sort of events where the system changes its state in an abrupt and fast way. And the cool thing about it is that, and this can be any physical system. And the cool thing about it is that these physical systems, when they switch from one state of the entire system to another state of the entire system, they have some early warning signals. So they signal that the change is coming. So in order to understand how it works, let's do the following. I want to see more. Let's see. Okay, good. Especially you, Niv and Anat. Okay, so take both of your fingers. I'm sorry, I want a positive, so bear with me. Take both of your fingers and move them together. Okay, small sync, little sync. Okay, increase the frequency, increase. Everything works fine, right? Okay, stop. Now let's do the opposite. Do out of sync. So one up, one down, and faster. Yeah, Shanat. So you see that you switch from out of phase into in phase if you went fast enough. Okay, so this is a kind of a bifurcation event, right? Switching from when you were in phase, it was stable throughout the entire frequency range. When we were out of phase and we started moving, then at some point the entire system switched to in phase. So and in order in order to understand better what is happening there, you can think about the system as existing in a sort of a potential well. So a stable state are these wells. Okay, this is a stable well at the in sync in phase state. Okay, at 0, and we see that there is another stable state here at out of phase at this up down. And what happens is that as we increase the frequency of motion, this well becomes shallower and shallower. Okay, at one point, the entire well is sort of removed, and the ball flows into the only stable state that exists, which is the in phase. But let's think what happens to the ball. The ball is the system, okay? Is the way we move. So what happens to the ball before this well is erased? So before it is erased, it's very shallow, okay? So when it was in the deep well here, it was moving fast. The autocorrelation is going to be super low, even it could be negative, okay? But when the well is shallow before it's erased, okay, this is still a stable state, okay. But the ball now can move really long trajectories, so the autocorrelation is going to grow really up, because if I measure the velocity of the ball going right, measuring it again, probably it will still go right because it has more value, okay. So the basic idea is that if you measure the autocorrelation and the decay time of the autocorrelation of the fluctuations of the motion, okay? It's not the motion is still at 0. But if you measure the autocorrelation of the fluctuations, you can see these early warning signals, okay? So let's start with the attacker. They have sensors on their finger, wrist, elbow, right shoulder, left shoulder, torso, and head. And this is a portrayal of their velocity trajectories. And what we see is that indeed, before motion onset, sorry, which is about 400 something millisecond, you see these spikes in the autocorrelation in lag 1 and the autocorrelation decay time. Okay, so there are some early warning signals, but are they related to attackers' motion? And the answer is yes. Okay, so it's quite noisy. There are 2,550, I think, points here, but 90% of them lie on this line. Okay. So there is a super significant correlation between the early warning signal and the attacker's motion onset, and indeed it preceded by about 130 milliseconds. So you can say, okay, good. The attacker indeed has this early warning signal, but maybe the blocker they read something completely different. Okay, I'll skip this. There's no correlation when you check only the finger. It's in the body. Okay, but let's focus on the blockers for a second. So blockers motion onset is also correlated with these early warning signals. So knowing the early warning signal, I can predict the timing of a blocker motion onset. So probably they do use this kind of signal. The cool thing about these kinds of predictions is that, and early warning signals, is that we can understand what kind of bifurcations. There are 3 different kinds of bifurcations. There's a pitchfork, hop, and saddle node bifurcation. And by the way, the variance diverges near the transition point, near the motion onset, we can infer which kind of bifurcation it is. Okay, So I can write a specific model for describing the motion of participants. If I have a model, I can do something else, which is a lot of people said, okay, you're right, you have these early warning signals and stuff, but people look on the gaze. There's something in the shoulder. There's something in their ear. It fluctuates really fast when they start to move. So what we can do is we can take a dot on a screen and make it fluctuate, either by using this mathematical model of how we think people move, okay, and the noise is correlated and going right or to the left. And I can do the same, taking a dot and using a non-correlated version and just going right or left. Okay, so one condition has these early warning signals, and the other condition does not. And what we see is, again, that people react by 100 milliseconds faster when you have these early warning signals. Okay, so even in its purest form of a dot on a screen, people probably know how to recognize and act upon these kinds of signals. I see I'm a bit out of time, so hopefully I managed to convince you that people know how to distinguish and act upon these early warning signals. And the question remaining still is, what is the inference mechanism? Right? People manage to do this task with 98% of accuracy, which is amazing. So what is the inference mechanism is something I would really love to find out more about. We have in the lab super advanced technology. We record markers from people's motion at a 1 millimeter resolution at a rate of we can know all the markers positions every millisecond and a half. So this is exciting, and again, that was me. Thank you, everybody.
Thank you very much, Yuval, for an excellent talk. Super interesting. We'll take one question. Anyone? Okay. Oh, did I see a hand? No. Anat Perry, go ahead.
Hi, Yuval. Thank you for the fascinating talk. What I was curious about, and I think I'm curious about it since I've heard versions of this talk before, is do you think that these cues are everywhere, or we just don't know where they are from the body, or each person signals it a bit differently? So you see these cues, but you see them also when you turn when you hide the head or hide the torso and so forth. So can we find them at the same frequencies from all over the body?
Yeah, so I think we do, and I think because it is a kind of motion that recruits the entire body. You can think about it as seeing the sort of the preparation stage of unlocking all the locks before starting to move. And because it recruits the entire body, I think you see the signal throughout the entire body. So what we did is analyze the first PC of motion, and if you look on the loadings, the principal component of motion, and what you can see is that the loadings are pretty much equal. So every part of your body contributes sort of the same amount of information to the entire motion. This is why each part of your body that you see is enough. Probably seeing all of them together brings more information to the system. Thank you.
Thank you.
Thank you very much. And moving onward to our last but not least talk. So David Omer will discuss space, time, and others in the brain.
Can you see my screen? Okay, so hello everyone. So my name is David Omer. For those of you who don't know me, I'm from the Safra Center for Brain Science at the Hebrew University. And today I'm going to talk about neural representation of space, time, for self and others. So it's a bit of a change of scale from what you heard so far. So I'm going to talk a little bit more at the level of neural circuits, more specifically in the hippocampus. So the study I'm going to present you today was done on bats. So this is kind of my previous work at the Weizmann Institute, and you can see coming up on the close horizon are marmosets, and I'll tell you a little bit about them and what we do with them in my group at the end. So, kind of stating the obvious, especially after we heard the previous talks, I'm sure you all agree with me that coordinated social behavior is crucial for survival and reproduction of any social animal. And I don't think you can even think about any example of a social behavior which doesn't require coordination in space and time in general. And obviously, somewhere in the brain, there must be neural mechanism to support this. The thing is that the hippocampus was never considered, and this is also again sort of mirrored from previous talks in this session. So the hippocampus was never really considered to be part of any system involved in social behavior, or to mention social coordination. And the reason for this is very, very simple, is because for almost half a century, which is a lot of time, the view of the hippocampus role in behavior was dominated by representation of self. And this is until very recently. So you can see here kind of the hallmark neural representation from hippocampal of rodents, like place cells and grid cells, which represent my position in space. But of course, there are other neural representations in the hippocampus, like head direction cells and border cells and object vector cells and many others. But again, the common denominator is that they are all involved in the representation of my position in space, and nobody ever considered the hippocampus to be involved in anything social. So about 4 years ago, we discovered this kind of new representation in the hippocampus, which we termed back then social place cells. Now we more think that this is a kind of a different facet of classical place cells, and I'll explain this a bit in a moment. So the way we discovered those social place cells that represent the position of others, is that we use this delayed match to place task, and I'll explain very briefly. So the task was designed for two bats. There was an observer bat and a demonstrating bat. And there were two phases for each trial. So on the first phase, the observer bat had to stay stationary on the start ball, and then watch the observer flying to either ball A or ball B, remember it, and on the second phase, he had to imitate the flight trajectory of the demonstrating bat. This seems to be a very hard task to sort of train bats, and bats are highly social animals. But because they are social animals, it turned out that it's very simple if you choose the right combination of a subordinate male and an alpha male. And maybe the most important thing I forgot to mention is that we recorded neural activity during the whole experiment from the observer bat using wireless neurologgers. So the bat could actually freely fly while we record neural activity in real time. So this is basically the result, and what you see here on the left, this is a top view of the setup. In gray, you can see the flight trajectories of the bat himself. In red, you can see dots representing spikes emitted from a single cell in dorsal C1 hippocampus of the bat that we recorded from. And this is a classical place cell. So this cell tends to fire when the animal is at a particular location. And this is exactly what people have reported before in rodents. So in principle, there's nothing new here. The discovery is what happened when the bat that we record from was actually stationary on the start ball. And this is what you see on the right. So when the bat was stationary on the start ball, the demonstrating bat was flying. So this time in gray, you can see the trajectory of the other bat flying either to ball B or ball A, and superimposed again spikes from the same unit. And you can see that the same unit not only capable of representing my own position in space, but it also represents the position of another conspecific animal. So we called this representation back then social place cells, but again, now we think that this is just another facet of classical place cells. So place cells in the hippocampus are capable of representing both my position, but also position of other in social context. And you can see here different examples, and we found many cells like this. So each row here is one example. On the left, you can see a representation when I'm flying, so it's a classical place cell representation. On the right, when the other bat is flying, so the same cell representing both my position and the position of another bat. So going back to the stated obviously, so as I said before, almost every social behavior requires coordination in space-time, and most probably those social place cell representation play important role in those social behaviors. But as I keep mentioning, social coordination requires representation of not only space, but also time. And this is a different, completely different dimension. And this is basically the essence of the study I'm going to present to you now. So hippocampus is known for almost 20 years to represent also self-time, and in the form of what Pastalkova and Buzagi termed back then hippocampal time cells. So time cells in the hippocampus are, maybe first I'll describe the actual task that they help them to discover those representations. So they train rats to run in this 8-figure maze. And during in between the trials, when the bat was not running in the maze, it was trained to run in this running wheel for a fixed amount of time. And turned out, so obviously the animal is stationary, its position in space doesn't change. So it seems in a very superficial way that the only parameter which is changing is elapsed time from onset of the running in the running wheel. And surprisingly or not, they find many cells, each cell which with its own specific preferred moment of firing, and they turn them time cells. And even more importantly, at the population level, if you look at all of the cell, you can easily see, as you can see in this picture taken from the actual study, they form a very nice internally generated firing sequence over time, which theoretically can be used to decode elapsed time from the onset of the running in the running wheel. But there are two very important questions which were never answered in the context of time cell or hippocampal time cells. The first one is sort of loosely connected to the social context, but it until now it was not even known if time cell represent context-dependent experience or time per se. In fact, there's a sort of an arm race over the years to find neural representation of time per se in the brain. And so far, this arm race haven't succeeded to find such representation. And so when I say represent context-dependent experience, I actually mean that if every recorded neural representation comes along with a behavioral experience, it's almost impossible to tell whether those firing sequences represent the unfolding of the behavioral experience or time per se. And the reason it's very hard to dissociate between them is because it's very hard to dissociate between behavior and time. So I hope I'm going to convince you that we found such a representation. And lastly, the second question, which is more in the context of this session, nobody knows until recently how time cells code time in social context. So we set out to find time cells in the hippocampus of bats. We basically used the same experiment and the same data, but this time we focused on epochs when both bats were stationary. So focusing first on self-representation of time, we simply had 3 different conditions, as you can see in those cartoon here. So in each of the conditions, the bat himself, the bat that we recorded from, simply landed in one of the locations in the room. And as you can see here, we also find time cells in the bat hippocampus, not surprisingly. Let me walk you through this representation. So the x-axis is time elapsed from the landing moment of the bat himself. On the y-axis, you can see different trials And so each row here represents one trial. So the neural response of a single neuron of a time cell from the hippocampus. And you can see that these particular cells tend to fire very precisely at a particular moment of time. Hence, it's a time cell. And surprisingly or not, we find many time cells like this, like people find in rodents. And each one of them have a different preferred moment of firing, as you can see in this example, without going very deeply into the details. And at the population level, they also form very nicely those firing sequence that unfold when the bat is landing in one of the locations, as you can see here. And very interestingly, this the epoch of time, which is spanned by those firing sequence, turn out to fit exactly to the average time the bat himself spent in each of those locations, which is a strong indication that maybe those neural sequences represent elapsed time from the landing moment. But looking carefully into the actual representation of different cells, we found something very interestingly, which was never reported before. We found many cells which are time cells only in one location. So this is one example on the top, and you can see that this is a time cell which actually behaves like a time cell only when the bat is landing in one location, in this case, landing ball A, and it literally fires almost no spikes when the bat is landing on different, on the other two locations. And below you can see another example of such a cell. So only looking at those few examples, you can easily tell that those specific cells are not encoding only time, but also spatial context, also place. And it turned out to be that the majority of time cells that we found, as you can see, so this is a Venn diagram showing you the distribution of time cells that were recorded. And when I say time cell, I mean self-time cells. And you can see that in each of the location in the room. And you can see that the majority of the cells, the majority of time cells are tuned only in one location. And this is more clearly seen if you look at this other type of representation. So if we take all time cells which were found to be significant time cells in one location, in this case landing ball A, and we sort them according to the preferred moment of firing, we get this very nice firing sequence. But if you look at the same cells, now when the bat is landing on one of the other locations and apply the same sorting criteria, you can easily see that the firing sequence is almost gone. And this is true also for the other location in the room, for the groups of cells which are tuned in each of the other locations. So it seems that at the population level, time cells represent not only time, but also spatial context, which is in a way is very interesting because it immediately pops up the analogy to the concept of episodic memory, which is basically defined as a representation of what happened and when. And in a way, those groups of cells fall exactly into this category. So until now, everything is it seems to be in place, but now is the point that things are starting to get a little bit more interesting, because if you look carefully, even in those pictures, you can see that there's, so what I told you just a moment ago is that when the bat is landing on other location, the sequence is completely gone, but it's not. And you can see this in a very faint halo over here. So it turned out that if we look only at cells which are tuned on both locations, so let me remind you again, I was talking until now mostly about cells which are tuned only in one location, hence they were coding time and position simultaneously. But now I'm looking at cells which are tuned on both locations. And this is what you see here. So the top row are time cells tuned in both locations, sorted according to the preferred moment of firing. And if you look at the same cells, when the bat is landing on landing ball B, it's a different location. You can see that there's a significant amount of cells which preserve their moment of firing. So in other words, there's a significant number of cells which are pure time cells. They seem to completely be agnostic to the position of the animal. And in a way, they most probably represent time per se. This is another way to quantify this. So if we look at the group of cells which are tuned in both locations and look at the distribution of the difference in the preferred moment of firing in the different locations, so the group of cells which are tuned in both locations is here in the pink distribution. centered around 0 and very narrow and very different from the shuffle distribution. So if you look at the same population of cells and we shuffle them, this very sharp peak is completely abolished, which basically says again, in a quantified manner, that cells which are tuned in more than one location tend to preserve the moment of firing. And if you think carefully, the sorting criterion for this group of cells, as I told you, was cells which were tuned in more than one location. So this criterion doesn't say anything, and we shouldn't expect those cells to preserve their time tuning. And this is exactly what I just said in a moment ago. There's no reason to expect that time cells which are tuned in more than one location would show similar time preference in different locations. But again, another important question is, so we found these two distinct. We found those two populations of cells. Do they represent distinct populations of cells? And it turned out that this is exactly the case, as you can see here. So what you see here is this distribution of the correlation coefficient between the average firing pattern of each of the cells which were tuned in both locations. And you can see the distribution very skewed towards higher correlation values. And it's very different from the distribution of cells which were tuned only in one location. So there's a very nice bimodal distribution, which strongly suggests that what we see here are two distinct cell populations. One population, which we termed contextual time cells, which code for time and position simultaneously, and the other one, pure time cells, which are called time per se. So going back to the second question, which is more relevant to this session, how do time cells encode time in the social context? So we use the same experiment, and this time, and again, we recall it from the bat, marked here with a red log here on its head. And so in those three different conditions, the recorded bat was always stationary on the start ball. So the only thing is changing between different conditions is the position that the other bat is landing in the room. So the other bat is landing either on ball A, B, or the start ball. And lo and behold, we also find time cells which represent time for another bat. So this time, the x-axis is elapsed time from the moment of landing of the other bat. The bat that we recall the spikes is stationary on the start ball and is completely motionless. And you can see that we find many cells which represent elapsed time for another bat. And at the population levels, also form these very nice time sequences like regular time cells. And also here, if you look carefully, you can see that the time epoch, which is spanned by those time sequences, seems to fit very nicely to the average time the other bat was spending on each of the locations in the room. And we also have some evidence that also time cells that represent time for another bat are composed of two different groups, contextual and pure time cells. And obviously the numbers of cells here is much lower, but this is highly significant. So to summarize the three main results I just present to you, so we found two distinct populations of time cells in the bat hippocampus, which is completely novel. And they basically represent two different time codes to represent time in the brain. More importantly, we found a very specific and special group of cells which we termed pure time cells, which seem to encode time per se, which were never reported before in the brain ever. And obviously those cells can be used to code interval timing. And finally, maybe the most interesting result is that we found that time cells can represent time for another conspecific or another individual. And this is basically sort of the final piece in the puzzle of neurocircuits which represent both space and time for other. This is basically, again, stating what I just said in a moment ago. So those different representation of social places and time cells for other can support or probably support coordinated or synchronized behavior, as people here prefer to call this. So before I tell you a little bit more about what my group is focused now, I'd like to acknowledge the people who were involved in this studies, which I did at the Weizmann Institute in Nachum Milanoski Lab on food bats. So this is the main question that my group is actually focusing at the moment. Is there a cognitive map of social space in the brain, or more specifically in the hippocampus. So place cells are considered to be the physical embodiment of a cognitive map of physical space. So a cognitive map is basically a way to represent an abstract space in a relational manner. So we hold information about a distance in any space between each node in this map. And what we are interested in, there are some evidence that this might be the case for the hippocampus, is there a cognitive map of social space in the brain? And this is obviously something which we humans are very good at. And this is mainly the reason why I decided to switch to a completely different animal model than bats. So this is the animal model that we are using in our lab. Those are marmoset monkeys. They are no bigger than an average grown rat, as you can see here. And they are unique in particular for the questions that we are starting to answer, is because they have a very highly complex social behaviors, and they have a very similar structure to human in terms of a dyadic family. And maybe most importantly, they have they have a primate brain, which is a completely different machinery than a brain of rodents. And what we do is we use, again, wireless neurologgers, which enable us to record neural activity in real time while animals are naturally behaving. So for social behaviors, this is obviously a very important features. Along with the neural activity, we can record also audio signals, but most importantly, and this is when primates comes in, is we recently developed a new methods to record eye gaze from freely behaving animals, these marmosets, while they are freely socially interacting with others. And this, of course, obviously puts us in a position to study specific behavior which might be related to cognitive mapping of social space, like social gaze and eye contact in non-human primates. And for those of you in the audience who are interested in position of postdoc or PhD, feel free to contact me, and I will stop here and I will be happy to take questions.
Thank you very much, David. Super interesting talk. I already see one question. Yes, Ayelet?
Hi, thanks for this really interesting question. Sorry. Interesting. I have a question about your fascinating talk. Sorry. So you're basically showing that you have place cells and time cells for the other. And I wanted to hear you hypothesize what might be a neural computational principle for separating the two. So obviously the animal somehow knows that this firing rate is associated with self-location, and maybe another firing rate is located with some other conspecific location and also time. So what are your thoughts there? Does it have to do with phases of Campos theta? I don't know if you have, I guess you don't really in the bat, right? So some hypotheses there, I'd be very interested in hearing.
Well, okay, so I'll start by saying that, so this is a very general question in general in the brain. I mean, how do sort of the homunculus question, how do one region knows to read the incoming very rich information coming from multiple neurons, and katonti to suggest new ways, because the people have already dealt with this question and suggested many different mechanisms, as you alluded also, neural synchrony and frequency coherence and all those of kind of time mechanism. But I would put, if you ask me seriously, I would put my bet on something completely different. So I'm an avid believer in the fact that single neurons are important, but more important is the cell population. And there are also some studies suggesting that, for instance, the study from Yaniv Ziv, that the actual identity of a single cell is not the real important fact, and what's important is the whole state of the population. And so I would put my money over there. I don't have a very good hypothesis to tell you what would be the exact feature, but I think that's where the money lies.
Thank you.
I also have a question on that topic, if I may give myself. So if you talk about cognitive maps of social environment, or if you talk about cells that calculate time or space for self versus other, do you think that the calculation goes first to the self or the other and then inferred to time or location? Or do you think that it's the other way around? Maybe the brain just makes a cognitive map of the environment, and for some animals the environment is social, and then they become experts in making social maps, but the calculation or the mechanism can be the same if the environment is not social.
So I'm not sure I completely understand your point, but I'll try to answer. So I think I recognize two different questions what you asked. So the first one, it seems like there's a common denominator between social place cells, time cells, and time cells for the other. And even if, and this becomes like an obscene word in neuroscience, and even mirror cells. And this common denominator is the fact that you use the same neural circuit to represent both self and something for others. So this, and this was a very, very interesting idea that believers in mirror cells back then tried to push very hard, and it, by the way, it completely collapsed. And in a way, social place cells bring this idea back into the scene, because unlike mirror cells, when you have a very small number of congruent cells that code for the same thing when I'm doing something, when the other one is doing the same thing, and theoretically, this kind of representation will enable you some sort of an understanding, because you're using the same representation to represent myself and also represent others. So while this notion completely failed with mirror cells, because the number of congruent cells was very low when it comes to mirror cells, with social place cells and time cells for the other, it comes again, because maybe what you are sharing is not the same neurons or nodes of the map, but what you're actually sharing is the cognitive map. So it's the same cognitive map that you use to represent either me or self.
But doesn't that actually say the opposite? Doesn't that actually say that the map represents the feature, and then I can use the same map to represent myself or the other? I can apply it differently by the context, but the map just is doing something different. Like that's.
Well, I would say that the map is doing the same thing. It's
it's doing the same thing, but it's the same map that can be applied to myself and to the other.
Exactly.
Yeah.
And I think this is the congruency that sort of saves social place cells from this fallacy of mirror place cells. So mirror cells failed this concept because the number of congruent mirror cells is very, very low. So if you fall back into this, if you go one level up and suggest that what is the common denominator for self and other representation is the cognitive map, then you're basically using the same structure or the same neural network to represent self and others, and this is what maybe enables you to understand others. But I have to say that this is not on the verge, but this is literally waving hands. It's very interesting to discuss those issues, but we are very far from even saying anything close to such ideas at the moment.
Thanks. Any other questions?
I think we reached the end of
yes,
session,
we have.
so maybe just say again thank you so much for David and Simon and Shir, and also I want to thank going on David.
Yuval, can you say again?
The ISCOP organization committee, Nuri, Shachar, Esti, Yuval, Dominic, for giving us both the place and time to present our symposium today. Thank you so much.
Thank you so much for a beautiful symposium, and the poster session is already out there, and there's another symposium session at 1:30. So see you later. Thanks, everybody.